Hello, everybody. We're going to get started here. If you don't mind, just coming on in, grab a seat. All right, we'll get going. My name is Matt Weiner. I work here at RAIN. Thank you all very much for making it out today. See a lot of similar people. We appreciate everybody who comes to each month. Thank you for all the new faces as well. Thanks for making it out today. It should be a really good one. Should be. Just to briefly tell those who haven't been here before what we are, a RAIN incubator, we're a nonprofit life science innovation hub. Our goal is really to just grow jobs, companies, and talent in biotech for Tacoma. I want to take a quick second to thank all of the sponsors who help make Superheroes of Science as well as the rest of RAIN possible. Our community partners as well. Girls Who Code is starting up for our second year very soon. Still open for applications. Please check out the website if you're interested in joining. We're also still looking for mentors, so if you'd be interested in helping out with Tacoma's Girls Who Code Club, we would really appreciate it. Next month, Superheroes of Science, be October 14th. Be very different, very interesting as well. It's going to be all about the role of specifically coyotes and raccoons in urban ecosystems by Chris Shell, who's an assistant professor down at UW-Tacoma and a very good friend of RAIN's. With that being said, I won't hold you any longer. I'll welcome Dr. Stanley Langevin to the floor.
Thank you guys all very much. Obviously, I know a lot of you here. I have been a part of RAIN. This will be my fourth Superheroes of Science. So I've done quite a few of these. I feel like I have kind of seen RAIN from its inception in David's eye about building a biotech incubator in Tacoma. And when he first told me this story, that he was moving back to his hometown to do this, I was always very intrigued. When I moved to Seattle, that's where the connection grew stronger, and I started coming down here because of David. And the more I came down here, the more I realized this is the place that I need to be. So I have put a lot of my time and resources into not only being in Tacoma, but trying to assimilate into Tacoma both my career and my dreams. So this being said, I spend a lot of time at RAIN. I try to teach people here and the students that come in. I've now started teaching over at University of Washington, Tacoma, and the students there. I did a microbiology class this summer. So now that I'm faculty there, I really want to more embrace what Tacoma has to offer and try to work with David to try to combine some of these great resources that are down here and see if we can really pursue research in Tacoma rather than just teach. So that's my spiel. Without further ado, I will now get into my talk, which is kind of a culmination of the research that I've done and kind of how I have come to terms with how we view disease, especially infectious diseases, and how we can, in the future, learn how to treat them better, because it's not just about the pathogen itself. There's a whole ecology that surrounds that, and that's going to be basically the talk. So I'm going to first go into Postulates. So this is the idea of one pathogen causing one disease and the germ theory. And then I will talk about some of my experiences applying that germ theory to human pathogens and the fallbacks to those and what I learned. That then led me to the model that I am now pursuing and a lot of researchers are pursuing as well. Then I'll get into what is the pathobiome, and that is what I believe is more or less the future and the replacement to Postulates. And then I'll also talk about how these systems-based approaches can be used to monitor health and prevent disease. Okay, what are Koch's postulates and the germ theory? So the germ theory is best described as one pathogen, one disease. So when I refer to a pathogen, I'm referring to the classical definition, which is a microorganism that can cause disease in a specific host. When I refer to disease, I'm referring to a condition in which the host is harmed because a group of host cells or tissue is unable to work. And that's usually in conjunction with a pathogen. So here are the players. As far as the pathogens, a lot of the pathogens that we are aware of are ones that cause severe disease. That's how they get labeled. So things like Ebola virus, influenza virus, Salmonella, and Plasmodium falciparum that causes malaria. These are all pathogens that are classically known to cause human disease, and they are pathogens that, when present in humans, is usually associated with a particular disease. And these are the infectious diseases. When you talk about diseases that are caused by pathogens, you can have a range of symptoms, and it depends mainly on the pathogen itself and its tropism to what it likes to infect, and where it likes to go in your body once it gets in. So the route of entry is one, and then also the cells that it likes to infect. And these can cause a range of diseases, everything from encephalitis, where viruses that are neurotrophic and get into your brain can cause disease, everything from acute respiratory diseases, which can be caused by bacteria and viruses and fungi, to hemorrhagic fever diseases, which basically cause your immune system to overreact, which causes a lot of the disease manifestations that occur, to gastrointestinal diseases. And these are a lot of enteric bacteria and viruses that can cause these type of diseases. So the pathogens and the diseases. So what did Koch do? So Robert Koch was a scientist in the late 1800s that was one of the first scientists to really further the Louis Pasteur's idea of one pathogen, one disease, and the germ theory. So he was able to prove that Bacillus anthracis, a bacterium, caused anthrax. So by being able to do that, he came up with a series of steps that he then coined Koch's postulates. And these 4 steps we've been using since then to be able to confirm that a particular microbe is causing a particular disease. And in order to do this, you really had to demonstrate this almost every time. What are they? So Koch's postulates is basically based on 4 simple steps. The steps are: you observe an animal or a host that has a particular disease. What you need to do then, and you find it in every case, you find a microorganism associated in every case that causes disease, but not in the healthy host. Then you have to basically isolate that organism and grow it up. So then you have to be able to confirm that it is a bacterium, a virus, a fungi. And then you then have to take that microbe that you grew up and cultured, and you then have to put it back into the same host to be able to demonstrate the same disease symptoms as you saw in the original host. And then once that, then you have to be able to pull out that microbe again out of that second host to determine that that microbe actually caused the secondary disease. Now, once you have done those 4 steps, you have fulfilled Koch's postulates, and now you can actually say that this particular pathogen causes this particular disease. And then it goes in all of our books, and then we all read about them, and we go, hey, this is the virus, or this is the bacterium that we need to look out for if you are showing these symptoms. Okay. So now I'm going to go into kind of my experiences. So I did a lot of infectious disease research, outbreak research at CDC, going around and trying to figure out how viruses are transmitted and how viruses are spread. Then I spent my graduate school days working with highly pathogenic viruses in different host species, and trying to figure out how different hosts actually relate to different viruses and the way that they cause disease. And then I moved out and then basically moved on to taking more novel techniques now that we have to be able to characterize all the microbes and the host response at the same time, giving us a more complete picture of really how disease is really formed. And that's what I'm going to go into. So I first started doing, and this is in graduate school, doing research with West Nile virus, which is a virus that first came to the United States in 1999, and when it did, it caused huge mortality in corvid species, up to 100% mortality in some of these species. So we were wondering, okay, what's going on here? You have a virus that was never seen in the Western Hemisphere. It doesn't normally cause disease in birds where it's found in Africa. So why, when it comes here, does it completely wipe out these populations? What we found is that when we looked at the strain that came to the United States versus some of the other strains that are found in the world, that some strains killed all the birds and some strains did not. So we looked at this a little bit further to figure out, okay, is it a different West Nile virus or is it the same West Nile virus? And what we found is that we could take a virus that killed the birds 100%, and if we changed one particular amino acid out of 1,300, we were able to make the virus not kill the birds. So what it began to tell me was, well, this is the same virus. All we did was change one amino acid, and by doing that, it's no longer virulent and it's no longer pathogenic. So, but it's still West Nile. So now it's not really about the pathogen, right? It's about really understanding the genetics of the pathogen, and not all the pathogens, just because it's named West Nile or influenza, act the same at all. So that started to break into one chink into Koch's postulates, which it is one pathogen, one disease, but not all pathogens, even if they're the same virus, are the same. So then I moved on to do HIV research in Africa at Harvard. And this is where I was introduced to the host. So I was all focused on the virus and how the virus infected the host, but I wasn't really spending enough time to understand how the host response was causing or preventing disease. So by doing HIV characterization, we were able to discover that in order to fight the virus, we needed to understand both the microbe, then our body's response, and the human interactions that spread it. So this also got me into understanding what is disease and what causes disease. So I thought we get the virus or the bacterium, then it replicates at high rates, and it then manifests into the disease that we then get. But then I also started digging into it, realizing that, well, that's not necessarily the case, because by doing pathology, we've realized that the pathogens actually cause such a robust immune response and an inflammatory response that actually it's the host that ends up causing a lot of damage, and not necessarily the pathogen at all. And where this came into is that you have certain immune responses and certain inflammatory responses that are protective and some that are detrimental. And that we find that the longer these responses are turned on and activated, the more likely they're going to be detrimental than protective. So this gets into understanding, and this all came into understanding HIV and SIV in its natural host. So when you look at disease and you look at where HIV came from, HIV was circulating in monkeys for centuries. And basically when you look at the monkeys that circulate, that are the natural host to the virus, the virus is really in high loads. So it's replicating like gangbusters. But what's not happening is the host response of those monkeys isn't going off. So it's almost like the virus is able to replicate and do its thing, and the host response doesn't react. And in the end, what the end result is there's no disease. So they go their whole entire lives with high loads of SIV circulating in their bloodstream, but they never show symptoms of disease. Now you take that same virus and you stick it into a rhesus macaque, which would be a non-natural host to that, and it shows full-blown symptoms of AIDS. So what it really is getting at is that it's the natural host and the genetics of the natural host that are really driving disease pathogenesis and disease severity. So that's getting back to, is it really the pathogen that we're really looking at? Or do we really need to better understand how our response is acting to that pathogen? And so you can find that in natural hosts, the circulating pathogen is really high, inflammatory response and the immune responses are really low, and therefore the pathogen infects it for life without showing any disease symptoms. So, and this is also getting into when I started studying highly pathogenic viruses like influenza and realizing that it's not the lethal pathogens. They don't kill you. It's you that kills you. So figuring out that the host dies from damage due to an overstimulating immune response and an inflammatory response rather than the pathogen itself is really getting then back to what is really causing disease. So what about the microbes? Do they have an impact as well? So we know that the pathogen can change very subtly and change its ability to cause disease. We also know that the host response and the actual host genetics play a role in causing the disease. So we also looked at, do the microbiome and the microbes that live on us, the resident ones, are they associated with disease severity? So we did a study where we looked at microbiome communities and microbes in children that were infected with influenza. And we tried to say, if we swab their nose and look at their microbes, can we predict which children will have mild influenza versus severe influenza? And our pilot study was able to show that we could take children who presented within 48 hours symptoms of influenza, have no prior antibiotic use, and swab their nose and be able to use the microbes in their nose to predict if they were going to have a mild influenza infection or a severe influenza infection. And this was based on multiple years of influenzas. We were able to confirm this in the adult population as well. And what we found was a lot of people were like, hey, most infections that deal with the microbiome, that affect the microbiome, are when you have a respiratory infection, you're basically going to perturb your upper respiratory tract microbiome, and the players that are in your upper respiratory tract are then going to go into your lower respiratory tract. And depending on who's in your upper respiratory tract will then predict on if you have pathobionts or pathogens that are in your upper respiratory tract, you're more likely to get severe disease rather than just mild disease. So this really leads me to then push this whole concept of, okay, it's not just the pathogen, it's the host genetics, it's the microbes that live on us, it's really this whole entire ecosystem. And then also realizing that the environment also plays a role, which then we developed kind of a new way of thinking about it, a new paradigm in which you take in the host genetics, the host resident microbiome, and the invading pathogen, and that's how you really look at what the disease state really is, and then the outcome to that. So I think in the future, this is kind of where we're going, to take a more holistic approach to really understanding pathogens and how they cause disease. So this is really the systems biology-based approach. that I think is really needed to really solve some of these complex problems, because we are looking at a whole entire ecosystem involved with our host, the resident microbes, and the environmental stressors. So how do we study this? By then taking all this big data, and we have to be able to use computational analysis to better understand it and interpret it, and then hopefully we can come out with the right diagnostic and prognostic tools to then use in the clinic. So I think that this is where we're all going. Genomes are being sequenced. We are now using these genomes for more personalized medicine to better understand how we respond to certain therapies and to certain diseases. So I think that the next layer will just be adding the rest of the ecosystem, which is the microbes. So by incorporating our host genetics, we can predict the outcome to pathogen exposures. So since we have our genome sequenced, now we can then mine those genomes, and we can identify SNPs that are involved in different disease manifestations, and then we can track those. So you can then basically, before you even show that you have symptoms, we can already demonstrate that you would be more susceptible to a particular pathogen or a particular disease. And they were able to do this recently with malaria, where they were able to do GWAS studies and look at genetic polymorphisms, or SNPs, that are found in our genome, and they associated particular SNPs with disease severity to malaria infections. So this gets into looking at us as the way that I visualize us now, which is we are more microbe than we are human. So these genetic differences can play a role, but we're very similar to each other. So those variations, if they're playing a big role, imagine what the microbiome is doing, since we are 5 times to 10 times more microbe than we are human. I think it really puts in perspective to understand that when the majority of the genes and the metabolic processes that occur in our body are not human and they are microbial, we really need to rethink who is really in charge and who is really important in understanding our susceptibilities to disease. Because the further we go down this road, when I started doing microbiome research, I guess it was like 7 years ago, I would sit up here and everyone would be like, we don't understand, or this is just anecdotal. And as more research came out, from every angle, from acute infectious diseases to chronic diseases to psychological disorders, these are all being associated with the microbiome. So to take a step back now and really look at this one pathogen, one disease, knowing that really those pathogens are actually interacting with the microbes before they even interact with our host cells. So this is really the reality that we deal with, and this is, I think, that the future of medicine will really unfold, is really trying to get at this true ecosystem and understanding why, if I get exposed to influenza and you get exposed to influenza, you might go home and I go to the ICU. It's not the pathogen. So when I talk about the host microbiome, who am I talking about? A lot of times when people talk about the microbiome, they are strictly referring to the bacteria. That is not true, since the microbiome is composed of both bacteria, but also the bacteriophages, the viruses that infect those bacteria, which can be 100 times more abundant than the bacteria themselves. The parasites that also live in us, and also the fungi. So this constitutes the microbiome, and until we are looking at all of these players together as a community, we really, I think, there's a big elephant in the room that I think is missing from understanding health and disease. But the big problem is how do we really define who these bad players are? So by doing this research, we've also uncovered that we know who the bad players are. So when you get salmonella, or you get E. coli 870157, or you get Ebola, or you get influenza, we know what's going to happen. Those are the pathogens, the highly pathogenic ones that cause severe disease. And when you get them, usually you manifest certain disease symptoms. But what about this whole other layer of players that we are now calling pathobionts, that are bacteria or viruses that live, and they basically live as resident microbes in our flora. And then when the conditions are right, they don't cause disease, but when the conditions are right, they will cause disease. Things like Clostridium difficile, things like Pseudomonas aeruginosus. These are all things that even Streptococcus pneumoniae, 20% of us have Streptococcus pneumoniae in our upper respiratory tract right now, but it's not causing disease. So yeah, how do we understand and identify all of these players, which are now starting to come into focus now that we're able to actually sequence and visualize all the microbes? Because the big problem up until now, and why Postulates kind of failed in itself, was another way was that it only can look at things we can culture. Well, 70% of the microbes that live in our gut we can't culture. So by eliminating anything that we can culture, kind of eliminates a large proportion, I think, of the microbes that probably are causing some kind of disease, but we're not even tracking them. We're not even calling them pathogens. So understanding who is symbiotic, who's good, who's mutualistic, and then who's pathogenic. So I guess it was a couple years ago, the first group, I guess, started to adapt Postulates, is what they said, and to incorporate the greater disease complexity. So what they were trying to say is that we have to look at the microbiome, because we realize that the microbiome flora can actually resist disease. So that's automatically a big thing. And then they were also realizing that you can have co-infections. Like a lot of us are co-infected with multiple pathogens. So how does that manifest then in the disease that we see? It's not going to be the same as when you have the single pathogen. So really in the end, a lot of diseases, even complex diseases that we're seeing, like inflammatory bowel disease in our gut, is really the combination of host genetics, the resident microbes that are there, and the environmental impact. So if you are stressed, you're more likely to be susceptible to a particular disease. Hence why influenza likes to spread so well in the wintertime. It's not just because we're indoors, but we're usually when you're out in the cold, you have a weakened immune system. So these are all things that play a role in the fact that will you get disease or will you not? So this is kind of how I see it, more like the Postulates 3.0, where you have the microbiome, the pathogen, and you and your host genetics. That's forming pretty much the core of understanding if you get infected, what will then transpire. But then also incorporating the environment along with that. That there's pollutants, there's things that we eat, our diet. There's so many things that actually do affect not only our microbes that live on us, but also how we are feeling and being healthy. Okay, so I'll kind of start to finish things off with how I really think that knowing that it isn't one pathogen, one disease, and we need to take a more holistic approach into understanding infectious disease. How are we going to do it? So I think taking a more systems approach, or network approach is what I'm trying to call it, where you are using some of the more modern technologies to do both all the sequencing of all the microbes and your genome, to better be able to find out the correlations between the microbes that exist on you and the diseases that you experience throughout your lifetime, to be able to look at the biochemical, the communication between the microbes and your host cells, and then being able to come up with quick, rapid diagnostic, like point-of-care tests, even at-home tests, where we can better monitor this. Because a lot of chronic diseases are based on low inflammatory perturbations happening in your body over time, over long periods of time, and we tolerate these things. And I think that that's really what it comes down to, is that we all walk around acting like we're healthy, and all of us have something going on to where over time these things are probably leading to some of the downstream chronic diseases that manifest later in life, like cancers, autoimmune diseases, and some of the lifestyle diseases. So the systems approach, by doing it, by incorporating the host microbiome, the resident microbiome, looking at your immune response and your host response just as a baseline, we can start to then map out what does healthy look like for you over a range of time. And then by doing that, we can better define what it means for you to be healthy, because you being healthy and me being healthy might be different things. And then also the treatments that go along with that will be different. And I think that that's really where I thought that personalized medicine was kind of this word that was thrown around that was never really going to come to fruition for many reasons. But after doing all this research, I'm now realizing it's the only way we can do it, because to making the assumptions that we make right now, grouping individuals and saying we're the same when we're absolutely not, is really what some of the problems. I think we can get around understanding how drugs work and everything else if we can understand the whole entire community. And I think that's where it really comes down to. So we can also use all this information to better do diagnostics. So like we were doing a study where we were trying to figure out, if you come to the ICU with a respiratory infection, one of the first things the doctor needs to understand is what is causing the disease. And usually it's going to be bacterial or viral. And this is a very important decision that needs to be made fairly quickly, because it involves how the treatment will go. So if you can identify if a human being comes in and has a bacterial infection versus a viral infection, you can quickly start treatment and have an effect, because antibiotics don't work on viruses, but they do on bacteria. So what we did was we looked at influenza-infected patients and also humans that were exposed to bacterial infections. And we were able to not only look at and this is just looking at the host. This isn't even looking at the microbes. This is just looking at your host genes and what happens, what genes become activated when you become infected with a bacterium versus a virus. And what we found was there was distinct gene sets that were activated only during bacterial infections, and distinct gene sets that were only activated during viral. There were some that overlapped, but we were able to find groups that were very distinct. And by doing that, we can then make panels, right? So then the person comes in, you can just test the panel, and if they hit the bacterial panel, then they're more likely going to have a bacterial infection. If they hit the viral panel, then they're more likely to have a viral infection. And this really is now removing the pathogen altogether and just saying, I'm going to look at the host and use that as the diagnostic tool. So you can see how if we layer these things, we're going to have a lot better accuracy in being able to do. And once you have the panel, I can come up with quick, rapid point-of-care diagnostics using PCR-based tools to then look for it. So I don't have to do the sequencing and all this complex analysis to do it. So another way that we can control, so everyone has heard of CRISPR-Cas9 gene editing. So we're using CRISPR-Cas9 gene editing to look at a lot of genetic diseases in which we can edit the DNA of our genomes to then stop and terminate some of these diseases. But what I find interesting is that it's not just about CRISPR-Cas9. There's a whole slew of them that not only edit DNA but also edit RNA. So you can imagine if you have a situation where you get influenza or you get Ebola, and you know it's not the pathogen that's causing the disease, but the host. So what can we do? Well, if we can use CRISPR-Cas9 to edit the RNA genome, then we're not editing our DNA, right? So it's not a permanent change. All we're doing is trying to cut and block the RNA that's being produced of the genes that are causing the disease. So we can target inflammatory genes, we can target immune genes, and we can control them to where they don't overreact and then kill the patient. So instead of getting sick, you just get mildly sick rather than severely sick and die. So this would be eliminating the cytokine storm. We can also use, by doing all the sequencing of bacteria, we've been able to identify that the microbes are also controlling our host response with genes and proteins that they make. So we can actually use the bacteria genes as our own treatments to be able to control pro-inflammatory responses or specific immune responses. It also works well to use the bacteria genes to also activate B cells for vaccine efficacy. And the reason this is important in the end is that a recent study came out that it's not just about modulating the host, but we really need to understand the microbes because they are having a huge impact on the drugs that we take. So a recent study looked at like 68 different bacterial species that live in our gut. And they found that those bacterial species were able to metabolize over 271 common pharmaceutical drugs that we take. And not only are they metabolizing them, but they are changing them. So they're not even the same drug that you took. So this is really why when people say, you know, let's put everybody on Prozac, but Prozac doesn't work on a certain population, it could be because the microbes in their guts have basically changed it to where it's having no effect. So if the microbes are this important, not only for health and disease, but also the treatment of the diseases that we take, how do we, so I just described how we can try to control and monitor the host, but how can we control and change and modulate our microbes? Because I think that that's my next step. I mean, that's where I think all disease is going, is like, okay, we were able to describe the microbes in your gut. Okay, now what do? We don't really have any tools because we've only been trying to target and kill the pathogens. So all the other microbes that are now we have to be concerned about, we have no idea how to get rid of them, how to manipulate them. So we need to be able to really move forward with that kind of thinking. And one of those ways is using the viruses that infect the bacteria. So by using the bacterial phages, the virus that infect the bacteria, we can start to isolate those that are coming from bacteria that we want to target, and then we can use those to then just take out the bacteria of interest, rather than what antibiotics does, which is takes out a whole population of them. So we've been doing work with Betty Cutter down at Evergreen, and we've been sequencing some of the commercial phage cocktails that they use in Russia and Georgia, and they use them in the clinics now. I think that with the antibiotic resistance phenomenon that is now taken over the world, alternatives are really needed. I think this is really one that's being pushed now in the United States. I just hope it's pushed in the right way, not as a drug, but these are biologics, so they have to be treated that way. So yeah, so this work actually was done here at RAIN to sequence those guys. I'm also working on a project that is being done at RAIN, in which, this is also in collaboration with Betty Cutter, in which we're trying to, so if we want to be able to capture the phages of interest, how do we do it? So if there's certain microbes that I can't grow, can't culture, I can't get the phages out because I need those to be able to grow up the phages. So how do I do it? So we were trying to think of an out-of-the-box way to be able to collect phages without having the host. So the idea is to generate synthetic phage tails, or the tails are what they use to be able to attach to the bacterium. And then, so we can synthetically make any tail we want to target any bacteriophage we want, and then we make antibodies to target that tail, and then we can use those antibodies to then run any sample through, and if there's a phage that that is actually has affinity to that antibody, it's going to bind to it, and we can pull them out. Then we don't even have to be able to culture these things. We can actually isolate them straight from any sample, and then be able to characterize them through sequencing. And then now through synthetic biology, we can make our own phages. We don't actually even need them. We just need their sequences. Oops. Okay. That's going to be it for this talk. I want to thank Rain, obviously, who has been a great supporter of allowing me to come down to Tacoma and try to do research down here. And then obviously the University of Washington, the startup that I have here as well, Orneon Health, which is really trying to take a community approach to look at both the microbiome and your host response to better understand disease. And then the CDC as well, where I worked and did some of this work. Okay, any questions?
I have a quick question just to start it off. Sorry if I interrupted somebody. There's been some recent papers coming out about the interstitium and how viruses can kind of travel through the interstitium to get to different parts of your body. Do you think that the microbiome of the interstitium will be of kind of like a, importance in this kind of research?
Okay, so what he's actually talking about is, so this is a new bodily organ that we just discovered, which is pretty fascinating since I didn't think that would ever happen. So the interstitium ends up, I think, being it's going to be probably one of the most important pathways in which microbes both move throughout your body but also communicate. Yeah. There's no doubt in my mind. So I think that the interstitium is going to be an important pathway for understanding how neurotropic viruses get into your brain. I think a lot of these things are utilizing this whole organ that we really have never looked at. So my question is not necessarily not only characterizing what is in the interstitium, because it's really difficult, right? Because it's a fluid that's really hard to get at, and you get so little of it. But can we use it? Because I think in the end, it can be useful for something for monitoring, right? Because it's not invasive, right? And if you can do it, I could see how you could be wearing a patch that could then take microneedles and go into the interstitium, something and then can monitor. So the question is the interstitium fluid a useful sample type? That's what I want to know. And there are studies showing that it is, and it's comparable to the blood. And if that's the case, the blood. I think so. So yeah, the blood is a very difficult component to get. You got to use needles. It's invasive. So we need to come up with something else. And I've been trying. You can try to look at fecal matter. You can do saliva. There's only a certain amount of samples you can take from a human being or an animal. So finding this is a whole new arena of research and being able to use another tool to diagnose disease.
Hello. Here.
Okay, very good. Wonderful talk. I've got about 6 questions, but I'll ask one for now.
You can ask.
Okay.
Yeah, you can ask.
All right.
Later.
So most of your references regarding the biome and the pathobiome have to do with the oral path of administration of drugs. And so in addition to your work, what do you see happening in other pathways of administration? Inhalation, transdermal, intracutaneous, intraperitoneal, IM, IV, as a way to avoid the microbiome altogether?
Ooh, that's a really good question. I wish I had the answer to that. I think it's an immediate way to look forward, right? I mean, if you think about, I would look at it as the drug is only good if it finds the target, right? So administering your drug, how you administer it is important on where you're trying to get it to, right?
Right.
So I think that we could switch to other means as maybe a way to avoid it, but I haven't. It would be interesting to see a study in which they know that the gut microbes affect a specific pharmaceutical drug, and then they do a different route to see if it has an impact on individuals who are non-responders.
Similar question in the area of the biome. There's been some recent papers and also some recent development of compounds to try to first identify foods containing that category of offensive proteins called lectins, and which have been alleged to cause both IBS and also leaky gut syndrome, along with nutrients to bind those lectins so that they're not so offensive. And I'm wondering if that is a category of work similar to what you're doing in terms of things that actually affect the whole pathobiome.
Yeah, absolutely. I mean, I think that it gets more complicated, right? Because once you're bringing in, if you're bringing in specific allergens and ways in which the food affects our microbiome, that is something that I think is a very important part of research. I just personally don't know how to go at it, right? I don't know how to research it. To be able to, I think I could probably come up with a few ways to try to investigate it experimentally, but yeah, I think that it's going to be individual, and to be able to tie that with a particular microbe. I haven't really seen any data that's done that directly, right? I hope that answers your question.
You mentioned that there are gut bacteria that cannot be cultured. Why can they not be cultured?
Because we can't, because you have states in which it becomes really difficult to culture. So facultative anaerobic bacteria, they're hard to grow, right? And so are anaerobic bacteria. And the reality is that the complexity of, so when people try to do that, they can develop these microbioreactors to try to grow them up. And what we found is that they have to be grown as a community. For whatever reason, these guys do not want to grow as individual planktonic bacteria. And I think that that's really one of the main reasons why we can't culture them. Just the degree and difficulty on being able to develop the systems to culture them and being oxygen free, and then also the fact that they tend to grow up together. So like when these microreactors came out, where people are like, okay, we're going to take fecal samples and we're going to just grow up everything that's in that fecal sample, that's when we realized, because we could sequence it first and say, this is how many microbes are in there. Okay, now how many do you culture out? And by doing that, we're able to find that they were able to get like 60, 70% of them. So that's pretty good. But that still means that there's 30% not in that ecosystem. So until we can, when we have these experimental designs that are hugely flawed because there's elephants in the room, because we're excluding huge populations, I think it becomes difficult to find the real relevance, right? I think we create our own artifacts, and we don't understand it at the holistic level, which it should be understood.
Thanks so much, Dr. Langevin. That was a great talk. So I clearly see you think personalized medicine is a track for the future. But what is to stop us all from going on Amazon tonight and buying those kits that are going to help us track our microbiome and help us understand our personalized track? And do you suggest we look more towards startups that have this idea? What do you see
I
as the
think
future?
startups is a great idea to look at.
Any suggestions of
actually, I
which
think-
one?
Well, the bottom line is I think this is all too new. Like you have a couple companies that have been start to push this, companies like Ubiome, Second Day. What they're trying to do right now, the bottom line is we're at the cusp of being able to go from characterizing who's there to understanding the functionality of who's there. And that's what I've been driving towards, because it's like, okay, great. We know who's there. I can show you awesome color pictures of all the different microbes that live in different people's guts that associate with different diseases. But at the end of the day, until we know what the function of those individual microbes are in the community, then we don't really understand the microbiome altogether, because you and I can have very different microbes as far as their species are concerned, but they have similar functions. And as long as you need to understand the function, not who they are. So now I've moved away from, so now the way I envision us now is just a pool of genes. So there is no human genome, there is no microbe genomes that I'm sequencing. I'm just looking at genes and saying, okay, these genes are produced by these guys, and that's why they're in the community. They are playing a role because they make iron, or they, help digest carbohydrates, or they, and that's really, the genes are really what makes up the function. So I'm more moving towards eliminating understanding us as a genome, but as a conglomerate of genes that then make up what makes us healthy or what causes disease. So yes, I think that we're not there yet, because all the startups right now are just characterizing, and they're characterizing at a level that we cannot use clinically. When you only get to the genus level of a microbe, it doesn't help me. If they show you streptococcus, so what? You have streptococcus that lives in your normal flora, but if you have a certain streptococcus, then it's bad. So yeah, so to me, they just haven't gotten there, and that's what I'm trying to push. Like, I want to have a company that pushes these ideas and tries to put them in. So no, I wish there was, because I would be using it. Until then, I am going to build it. Nope.
How about now? So when a child comes into my office crying with a bulging eardrum with his parents, and I am tempted to prescribe an antibiotic because the parents want me to, even though statistically it's a virus causing that otitis media. When am I going to be able to use what you've talked about to say, this is a virus, antibiotics won't do anything other than feed the bacteria within him already? How far are we away from giving physicians that ability?
I would say we're probably within 5 years. I think that the amount of data that's coming out is insurmountable. Like, I can't even tell you how fast things are moving. Like, the amount of data we're producing, being able to utilize some of these things, these advanced algorithms and computational approaches like machine learning and AI. I mean, I am using machine learning. I mean, I am constantly cranking on trying to better characterize the microbes at this gene level, and even break it down to just a particular sequence. And if we can do that, then yes, then we can. We will be able to quickly be able to identify specific genes that are associated with profiles of diseases, and then be able to then give you a quick panel that then you could run either in the clinic, in your hospital, because there are machines that are already available there. You guys already run PCR-based methods. So yeah, I think this could be fairly robust and quick. As soon as the key is there's always the leap from discovery to use, right, to the application. So it's really about what I'm curious about is how quickly are we going to adapt to this new information? Like right now, it's coming so rapid fast, our ability to understand genomes. I mean, I'm working with Madigan right now doing a personalized genomics approach to soldiers trying to look at diet. And when we first started, I'm like, okay, yeah, we have like 200 different SNPs I can look at that are associated with BMI and associated with fat metabolism and things like that we can use as a scoring system to say if you rank really high on these SNPs that you are most likely going to be overweight and we need to try to do a different approach to your diet. We started, I said 200. Then a paper came out that showed if you really want to do it, you need 2.1 million SNPs. Okay? So that just like, overwhelms you because then you're just like, oh my goodness. And that's the way I think it's going to be for some of the more complex things. I don't think we're going to quickly come up with a panel that will do that then a clinic can use. But I think like what I showed you with the virus versus bacteria and understanding certain diseases and certain microbes that are present, I think we'll be able to do it pretty quickly. And give you guys more robust tools as far as, I mean, like when I get to look at this, I get to look at the gene response of all 23,000 genes, which you guys, when you guys look at it in the clinic, you guys are only looking at specific endpoint parameters of inflammation or certain things that are guiding you in that disease. But imagine if we can up that ante where you can actually look at all the genes and try to diagnose things based on the responses. It's in looking at signaling pathways rather than just endpoint proteins. And does that make sense?
What are some things a person can do to improve their microbiome without getting banned from the European Union?
Proteus, it's always a good question. To not get banned from the European Union, it has to be natural. So, I mean, that's what they're doing in the European Union, right? So they have the phage cocktails, so that's natural. I think there's going to be big changes when that comes to that. I think that when we get down to it, I don't know how we're going to, in the end, manipulate and modulate our microbiomes. I think that that's going to be, whoever figures this out, I know there's going to be lots of groups in the next few years that are going to come out with ways to be able to characterize it, like I said. But I'm really after the ones that are actually going to figure out a way to modulate it. And the things that I described to you these are all brand new things, the modulators, not the phages though. But I think it's going to be a combination of those. And I think it's just going to be, we're going to finally figure out how they communicate. And I think in the end it's going to be, we're going to have to use their genes and their communicators to stop them from doing things. I think it's going to be more of a less where, yeah, we control the immune system. So you get Ebola or influenza and we just say, hey, calm the immune system down, calm the inflammatory response down, then you will survive. Or saying things like, hey, you have, what I'm interested in is 20% of us are walking around with lots of viruses or bacteria that can cause disease. We're natural carriers, but we don't show symptoms. Why do we have them? Can we just get rid of them? Because I think that they are inherently, you being a non-carrier is a problem. You get influenza, now you cough, you are still spreading that Streptococcus pneumoniae around, which then someone next to you can be susceptible to gain. So why can't we use things like CRISPR and actually get rid of the microbes? Use phages, use things to just say, okay, even though you're not showing symptoms of Streptococcus right now, we know that it's in your upper respiratory tract, so we're just going to get rid of it. We're going to give you some phage, we're going to get rid of it, so then it won't cause problems in the future. I think that that's kind of where we're going to go to that type of modulation. But whether it is through the natural phages or our ability to block and manipulate the communication between the bacteria, it's going to be one of the two. I just don't know which one it is. Is that it?
And one more comment for our pediatrician friend. There is a Seattle startup named OtoNexus that has implemented and has a 510K on a new ultrasonic otoscope that can differentiate eardrum fluid as to whether it's viral, containing, or bacterial,
That's
with
awesome.
more than a 95% accuracy in 10 seconds. So you might get your solution this
Even
year,
faster.
this year instead of in 5 years. But I have a question for you. When you talk about the percentage of our genome that appears to be retroviral, there's probably a similar percentage that has to do with other bacteria, and I'm wondering whether that is the information source that transcriptase uses to build memory T when we have a particular antigen come into the body.
Wow, that's impressive. Yeah, I don't see why it couldn't be. I mean, first of all, you picking up, most people don't pick up on that. I think that's one of the most fascinating parts about the human genome, that 10% of our human genome is actually retroviral sequence, and it's not even human. And that they found out that these retroviral sequences are actually regulating our transcriptome and our cells, especially brain development. So to me, this is the fact that we assume that only the coding region of our genome, which is 10%, was the only thing that mattered, and that the other 90% was junk, I think was the biggest fallacy of all human genomics. And it's really, I think the dive down is to figure out what the rest of it's doing. We know it regulates, but how is it doing it? You know, the whole non-coding RNA is such a mystery to me. And I think that until we can grapple with that, and understanding why these, because they're there, and we know most organisms, if they don't need something, they get rid of it. So these are all needed, and we just need to understand what they're actually doing. I agree. I think it's really fascinating. I think it has a huge impact on us.
Okay, I think we're going to end Q&A there. Thanks, Stan. Yeah, feel free to mingle and ask him any questions.
Thank you.